Well, good morning. Thank you all for coming, and thank you for allowing me to present the findings of our research. I'm going to talk about 3 feeding trials that we conducted with the endangered Lost River sucker at Abernathy. My co-authors are Ann Gannum, James Barron, Kieslana Wing, and Josh Rasmussen. When I saw that I was the last speaker of the symposium, I realized I didn't have to be concerned about the length of my presentation. So I think it'll be less than an hour, but you'll be home by dinner, I'm sure. So let's see how it goes. So given the nature of this symposium and the subject of my talk, I'd be remiss if I didn't mention Dr. John Halver. He was hired by the U.S. Fish and Wildlife Service in 1950 as the founding director of the Western Fish Nutrition Lab, and he went on to literally write the books on fish nutrition. So a lot of what we know about fish nutrition is based on his work and is in part a product of the Fish and Wildlife Service. So I'll talk about the Lost River sucker, or the LRS. It's a lake-dwelling species. It's long-lived, relatively large. It eats small organisms and detritus. It was an important food source for Native Americans, including the Modoc and Klamath tribes, and for early European settlers. And at least one cannery was built for the species on the Lost River for processing the fish. It was also an important recreational species. In 1966, about 12,500 of these fish were snagged as they swim up the river to spawn. They used treble hooks attached to spark plugs to snag the fish. And this fishery was closed in 1987 due to population declines. So the fish is endemic to southern Oregon and northern California, and it was federally listed as endangered in 1988. Causes of declining populations can include habitat loss and degradation, overharvest, and loss of fish to water management structures. So why did we conduct this nutritional research with this fish? The 2012 recovery plan for the species states that artificial propagation may be needed to prevent extinction. We hope it doesn't get to this point, but if it does, we'll have some information on nutritional requirements of the fish. And of course, there are other recovery actions, including habitat restoration, but this is the one that we were focused on. So a nutritionally complete diet will be required to maintain this fish in captivity, just as it would be for any aquatic species. But there is no published information on nutritional requirements of the Lost River sucker outside of some information on what they feed on in the wild. So how do we do nutritional research? Standard methods have been developed. Generally, each dietary treatment is fed to replicate groups of fish. Fish are maintained in identical rearing units. These might be cages in ponds. These might be raceways, ponds, tanks, buckets, you name it. And generally, each dietary treatment is fed to 3 or 4 groups of fish. And equal numbers of similar sized fish are initially stocked in each of these rearing units. And they are fed their respective diets for a period of weeks or months or maybe even a year. And at the end of these feeding trials, we measure things like growth rates, feed efficiencies, survival, tissue composition, skeletal deformities, a host of different responses depending on the question that we're asking. And skeletal deformities were important in this research. And then based on statistical analysis of these data, we can make recommendations about the most appropriate dietary treatment. So a little background on the role of nutrition in captive rearing of fishes. Acceptance of a commercial diet dictates in part the ease with which a species can be reared. And what I mean is that species like salmon and channel catfish will accept these commercial off-the-shelf diets at first feeding, at their first meal, and that makes things a little bit easier. These are bags of feed that we're going to feed to steelhead at Abernathy at their first feeding, so we don't have to provide them with live feed. But in contrast, many species will not accept these commercial feeds, so we have to provide them with live feed. And often we'll use things like Artemia or rotifers. And production of this live feed is labor intensive and expensive. Nutrient composition can be variable. For example, Artemia are usually deficient in DHA and essential fatty acid. So if the fish that you're feeding these Artemia to require this essential fatty acid, then you have to enrich the Artemia with DHA or the fatty acid. And then supplies of Artemia can be variable. So in contrast, commercial feeds can be produced which contain any nutrient profile, and that's because there's a host of ingredients we can add to fish feed to provide the nutrient profile that we want. The tricky part is knowing the nutrient requirements of the particular species, which we don't know for the Lost River sucker. Commercial feeds can be stored for later use. But unlike live prey, commercial feed doesn't stay in the water column unless there's upwelling. And so this is thought to be a factor in why some species will not accept commercial feed. And supplies of commercial feeds also can vary. The diet that worked well in the current research was Otahimi. It's made in Japan, so we have to import it. And it looked like we weren't going to get it until about a week before this third study occurred. So things happen even with commercial feed. So a question that we'll always ask is, what can we feed this newly cultured species, this, in our case, the Lost River sucker? Can we use a commercial diet, a diet that was developed for, say, rainbow trout or catfish or bass? We'll also ask, what would be a good protein source for this fish? Can we feed it vegetable protein such as soybean meal, or are we going to have to feed it fish meal or krill meal? And also, which lipid source is best for this species? Lipids contain different fatty acid profiles, and fish have different fatty acid requirements. So we take all this into consideration when we're looking at available commercial diets for a newly cultured species, or when we're developing new diets for that fish. So with that background information, our objectives were to obtain some nutritional information on juvenile and larval Lost River suckers, and to see if we could identify commercial diets that could be used with this species until diets are developed specifically for the fish. So in the first experiment, our question was, what should we feed first feeding Lost River sucker fry? I've circled a couple of the fish in this slide. I hope you can see them. They were small. So we had 3 dietary treatments. Those included live Artemia, and then 2 commercial diets, Otahimi and Hikari. And this slide shows the different dietary treatments. In the first one, we fed Artemia for 21 days, live Artemia to the fish for the first 21 days, and then we weaned the suckers over to the commercial diet at day 21. And then in the second and third treatments, we fed exclusively commercial diets all the way through with no live feed. So this is the experimental system that James Barron built that worked out real nice. Some people might call these buckets. And so we had 9 of these in the first experiment. We stocked 75 fry into each one of them. They were 10 days post-hatch. Initial mean length was 10.7 millimeters. The dietary treatments were assigned to 3 tanks of fish each, and the study lasted 6 weeks. So we measured weight, length, and survival, whole body total lipid concentration, and whole body fatty acid profiles. Since I only have an hour, I won't talk about the fatty acid profiles. And so we used the same statistics for all the experiments. We used one-way analysis of variance, except the level of significance was 0.05. And we used multiple comparison procedures to separate means when significant differences were detected by ANOVA. So here are the results. The letters at the tops of the bars indicate significant differences. So survival was significantly higher in fish fed the Artemia Otahimi treatment compared with fish fed the 2 commercial diets. So it looks like live feed is going to be required for these fish initially. And we also saw differences in weight gain. Highest final weight was in fish fed the Artemia Otahimi compared with fish fed the 2 commercial diets. And interestingly, whole body lipid levels were higher in fish fed Otahimi compared with fish fed the Artemia. So this indicates that I wonder what are the optimal lipid levels for this fish, given this information. Do we want a fatty fish or do we want a lean fish? And so to summarize, highest growth and survival was in fish fed live feed during the initial 21 days, but they did transition well to the Otahimi after that. And given the difficulty of rearing live feed, it would be nice if we can find a commercial feed that will work from first feeding for this fish, but we haven't found it yet. So in experiment 2, we asked which is the best dietary lipid source for juvenile Lost River sucker. We had 6 dietary treatments. The basal diet was the same for all 6 diets. The only thing that varied was the lipid source. We had marine fish oil, linseed oil, soybean oil, or a 1-to-1 mixture of each of these. 100 fish were stocked into each tank. Initial weight was about 0.5 gram, and the diets were fed to 3 tanks of fish each, and the study lasted 6 weeks. We measured percent weight gain and survival, whole body proximate composition, and whole body fatty acid profiles. At the end of the study, there were no significant differences in survival. So this suggests that any of these lipids would probably work okay for this fish. But we did see a difference in weight gain. Fish fed the fish oil, linseed oil combination had lower weight gain than fish fed the soybean oil diet. I don't know why the fish oil, linseed oil didn't work, if it was a palatability issue or maybe it was peroxidation of the fatty acids, I'm not sure. But generally, all of these lipids appear to work except for the fish oil, linseed oil treatment. Whole body lipid levels in this case were higher in fish with the highest weight gain or fish fed the soybean oil compared with fish fed the fish oil, linseed oil diet. So again, what's a good lipid level for this fish? I don't know. So to summarize, dietary lipid source affected growth and body composition, and several lipids or mixtures of lipids appear to be adequate for this fish. And information on whole body lipid concentrations is needed. So in experiment 3, we asked which commercial diet should we feed to juvenile Lost River suckers. We bought 6 off-the-shelf diets and we made 1 diet at Abernathy, the AFTC diet. The AFTC diet was made to sort of mimic what the fish might be eating in the wild. It contained copepods, daphnia, and black worms and some other ingredients, including vitamins and minerals. There was a range of protein and lipid levels in these diets. So 35 fish were stocked into each tank. Initial weight was about 0.9 grams. Dietary treatments were fed to 3 tanks of fish each. And the length of the feeding trial was 9 weeks. So survival was affected by the dietary treatments. The highest survival was in fish fed the Otahimi and the AFTC diets. Weight gain was significantly higher in fish fed the Otahimi compared to the other fish fed the other treatments. So the Otahimi worked well in this case. And we saw a significant amount of spinal deformities in some of these diets. It was as high as about 30% in the Cargill diet and very low levels in the AFTC or the Otahimi diet. And I'm not picking on the Cargill diet here. It wasn't formulated for this species. It was formulated for other fish. And so it and some of the other commercial diets do not appear to be a good choice for this fish. And this is a photograph of a couple of the obvious spinal deformities in fish fed one of the diets. And these are fish from another tank that had none of the spinal deformities. So what have we learned? Good growth and survival are possible in captively reared juvenile and larval Lost River suckers. First feeding Lost River suckers grow well when fed live feed, but will accept a commercial diet. And dietary lipid source can affect growth and body composition. Lost River suckers exhibit high growth rates, high survival, and reduced skeletal deformities when fed this otahime. And the fish are susceptible to nutritionally related skeletal deformities. What we'd like to know, the specific causes of these skeletal deformities, effects of hatchery feed on post-release survival, effects of hatchery feed on reproduction in captivity and in the wild, and the effects of nutrition on diseases of this fish. And with that, we thank the Klamath Falls Fish and Wildlife Office for funding this research. Scott Foote, Ron Stone, John Holmes, Jeff Poole, and Kelly Hawk helped with this research. So thank you.
In addition to these fish, you're trying to recover the fish, do they have significance for any of the local tribal groups? And if so, if the decision was made for artificial propagation, would the tribes do it? Would we do it? Any thoughts on those?
So it's my understanding that the Klamath tribe does have a research facility in which they are rearing the Lost River suckers. And as far as who would grow these for captive propagation, that's beyond my pay grade.
Mike.
I was just wondering, to what extent were you able to draw on previous information perhaps developed for June sucker, or what your thoughts on how this might be applicable to other lake sucker propagation programs?
We are familiar with the June sucker research, and they found that they found some skeletal deformities in their work, and it appears that minerals may be involved with the deformities that we saw here.